In this podcast episode, George Church, a pioneering figure in biotechnology, shares his perspectives on the rapid progress in biology, the future of human longevity, synthetic biology, gene therapy, biodefense, and the interplay between artificial intelligence and biological research. The discussion traverses decades of breakthroughs, current challenges, and visionary possibilities, offering a comprehensive view of where biology stands today and where it might be headed. Church begins by addressing the concept of escape velocity in aging, the point at which advances in biotechnology allow human lifespan to increase by more than a year for every year lived. He cautiously estimates that this could be achievable by 2050, given the exponential progress in understanding and reversing aging phenotypes. Unlike a sudden breakthrough, this progress is expected to be gradual, with people becoming healthier over time, rather than facing a binary outcome of survival or death. He emphasizes that while physical laws likely won't impose a hard limit, economic or complexity barriers might arise, though he remains optimistic. When asked about extending human lifespan to that of long-lived species like bowhead whales, Church highlights the potential of somatic gene therapy over germline editing, especially since billions have missed the opportunity for germline interventions. He envisions therapies that replace or rejuvenate cells, including the brain, through mechanisms akin to the ship of Theseus paradox, gradually swapping old cells for new ones while preserving memory and function. Although challenges remain, particularly in brain integration, he believes biology offers many levers that can be flipped to achieve rejuvenation. Church discusses the current limitations and future potential of gene delivery systems. While no existing technology can deliver gene therapy to every cell in the body, there are no fundamental physical barriers preventing this. Improvements in targeting specific tissues, such as a 100-fold increase in neuronal targeting by his company Dino Therapeutics, demonstrate rapid progress. He notes that therapeutic success often requires only a small fraction of cells to be modified, as some cells can produce enzymes or factors that benefit the entire organism. The conversation then shifts to de-extinction efforts, including the recent announcement of a dire wolf brought back through synthetic biology and ongoing work on the woolly mammoth. Church reframes these projects not as attempts to resurrect exact species, but as synthetic biology experiments aimed at ecological restoration and understanding phenotypic traits. He explains that millions of genetic differences exist even within species, and the goal is to identify the minimal set of genetic changes needed to recreate functional traits or ecological roles. This approach, he suggests, could extend to complex traits like intelligence, where a handful of genetic knobs might have outsized effects. Church elaborates on the balance between complexity and reductionism in biology. While traits like human height involve thousands of genes, some key regulators, such as growth hormone, have dramatic effects and are clinically useful. This reductionist insight allows for the development of targeted therapies and modular biological tools. He highlights the power of genome-wide association studies and multiplexed experiments to identify and test candidate genes or transcription factors that can induce specific cell types or phenotypes. This modular, engineering-like approach is central to his lab's work and the broader synthetic biology field. The discussion turns to the darker side of biotechnology, biodefense and the risks posed by synthetic biology. Church acknowledges the potential for mirror life or synthetic organisms to be weaponized, raising existential concerns. However, he notes practical deterrents, such as the self-destructive nature of some bioweapons and the ethical constraints on most researchers. He stresses the importance of surveillance, whistleblower protections, and societal consensus to prevent misuse. The challenge lies in balancing the rapid democratization of biotechnology with robust defenses and ethical frameworks. Church draws parallels to nuclear weapons, noting that while offense often has an advantage, humanity has managed to avoid catastrophic use so far. He advocates for realistic expectations about the dual-use nature of biology and the need for international cooperation, surveillance, and consequences for misuse. The conversation also touches on innovative strategies like recoding genomes to resist viral infections, which could provide new layers of defense, though offense will likely remain ahead in many respects. Despite monumental advances such as the Human Genome Project, CRISPR, and rapid DNA sequencing, Church reflects on why biology has not yet experienced an industrial revolution akin to Moore's law in computing. He argues that biology is now entering a phase of exponential progress, fueled by the convergence of AI, synthetic biology, and improved manufacturing techniques. The integration of AI with protein design, developmental biology, and materials science is poised to unlock new capabilities, including the creation of novel biomaterials and potentially room-temperature superconductors. Church highlights that biology operates at atomic precision in 3 dimensions, surpassing current semiconductor manufacturing in density and complexity. The arrival of synthetic biology has expanded the toolkit beyond traditional genetic engineering, enabling the use of non-standard amino acids and novel nucleic acid chemistries. These advances, combined with AI-guided design and massive parallel experimentation, allow for rapid iteration and optimization of biological systems, accelerating discovery and application. The conversation explores why protein design and nanotechnology have not yet triggered a revolution comparable to early expectations. Church explains that while AlphaFold and related AI tools have dramatically improved structure prediction, understanding and engineering protein function remains challenging. Functional validation requires experimental iteration, which is now feasible at unprecedented scales thanks to DNA synthesis and high-throughput screening. He envisions a future where biology and materials science merge, producing new classes of materials with properties beyond current electronics, such as biological polymers that conduct signals at the speed of light. The expansion of the genetic code to include dozens of non-standard amino acids will further diversify the chemical space accessible to biology, enabling the design of exotic materials and molecular machines. Church reflects on his work with brain organoids and connectomics, emphasizing the immense complexity of the brain's 100 billion neurons and 100 trillion synapses. While genetic diseases affecting cognition can be addressed with gene therapy, replicating or enhancing intelligence remains a formidable challenge. He compares replicating a brain to copying a complex book, where capturing the exact configuration is far more demanding than reproducing the genome alone. The entanglement of brain and body complicates efforts to engineer intelligence, and Church remains cautious about the prospects of creating artificial brains or inorganic copies. Nonetheless, he sees potential in incremental improvements and therapies that can alleviate cognitive decline or developmental disorders. The discussion touches on the possibility of biobots, biological machines capable of rapid replication and advanced functions like radio communication or even nuclear power. Church acknowledges the challenges, particularly the incompatibility of biological systems with extreme conditions like those in fission reactors. However, he points out that biological organisms already extend their replication cycles through environmental modifications, such as nests or habitats, suggesting that hybrid systems combining biology and engineered materials could emerge. He envisions a future where biological and mechanical engineering converge, enabling the growth of complex devices and materials in ways that complement traditional manufacturing. When asked about the likelihood of life elsewhere in the galaxy, Church emphasizes the difficulty of proving either the uniqueness or ubiquity of life. He suggests that laboratory demonstrations of simple pathways from inorganic molecules to replicating cells under prebiotic conditions would strengthen the case for life's commonality. He also notes that many space missions have not been designed explicitly to detect life, and that moons with subsurface oceans like those of Jupiter and Saturn represent promising targets for future exploration. Church remains open to the possibility that life elsewhere might use different biochemistries or genetic codes, and he anticipates that biology on Earth will continue to evolve and expand its molecular toolkit over the coming centuries. Among the technologies Church feels are underhyped is genetic counseling. He argues that while gene therapy is crucial for treating existing conditions, genetic counseling offers a cost-effective way to prevent many inherited diseases before birth. He highlights examples like Dwarkesh, a community-based program that has successfully reduced recessive genetic diseases through informed matchmaking. Church stresses that genetic counseling is not eugenics when it respects individual choice and autonomy. He believes wider adoption of such preventive measures could dramatically reduce the burden of rare diseases, improve public health, and alleviate economic costs associated with caregiving and treatment. Church expresses greater enthusiasm for AI tailored to scientific discovery than for general language models. He cautions against rushing toward artificial general intelligence, AGI, without robust safety frameworks, noting the ethical and societal challenges involved. He envisions a future where AI accelerates biological research by guiding experiments, optimizing designs, and integrating vast datasets. But he remains skeptical that AGI alone will revolutionize biology without careful stewardship. He also reflects on the limits of scaling human creativity and collaboration, noting that some biological problems may not be easily paralyzed or accelerated simply by adding more intelligent agents. Looking ahead, Church envisions a world where biology and AI co-evolve, leading to near-perfect health and enhanced human capabilities. He stresses the importance of safety, ethics, and international cooperation to ensure that these advances benefit society broadly. While challenges remain, the convergence of exponential technologies in biology and AI offers unprecedented opportunities to transform medicine, materials, and our understanding of life itself.